Then the next thing that we'll discuss is the morphology of chromosomes. But the gross morphology that we'll discuss. So the next time we will discuss the morphology of chromosomes. If we'll study the morphology, the best stage to study the morphology of any chromosome is metaphase stage. Metaphase stage is the best stage. So metaphase stage is best stage for study of the structure or for morphology of the chromosomes, while anaphase stage is best stage for study of the shape of the chromosomes. These two are very important. Now, why metaphase stage is the best stage? Because at this stage the chromosomes are much condensed. They are also small in size and easily visible. That is why this is the best stage to study. Now, if we study the morphology of a chromosome, then we will find that longitudinally it is split into two chromatids. Each chromosome longitudinally splits into two chromatids. That means this is one chromatid. This is chromatid number suppose 1. Then this is chromatid number 2. Chromatid number 2. That is longitudinally it consists of two chromatids, and these two chromatids, they are known as sister, or we can tell them as twins, because initially in the interface there was a single chromatid and during S phase it has doubled. So it is one is duplicate of another. That is why they are known as, we can call them as sister chromatids or the twin chromatids. Both are exactly identical. And depending upon the position of this, they are completely separate from each other, except at a point. This is known as centromere. This is known as centromere, or this is also known as primary constriction. Primary constriction. And the two sides present around the centromere, one is longer, the other is smaller. So usually the smaller arm is designated as P, while the larger one is designated as Q. The smaller arm is designated as P and larger arm is designated as Q. Now the centromere, at the centromere, the two chromatids join together due to the presence of a sticky protein known as cohesin. Cohesin is a protein, and due to this both the chromatids are attached together. Now at this centromere, which is known as primary constriction, here at this point there is no nucleosomes. There are no nucleosomes, then no histones. No histones, and it also remains uncondensed. And on either side of the centromere, there are two disc-like proteins. There are two disc-like proteins like this on either side, and they are known as kinetochores. Kinetochore. Now the kinetochore is the disc-shaped protein to which the spindle fibers are attached during cell division, so that the two chromatids move towards opposite poles. So this is the first one is the chromatid, the second one is the centromere or primary constriction. In some chromosomes, some specific chromosomes, in addition to this primary constriction, there is also a secondary constriction. Like this. So this is known as secondary constriction. Secondary constriction. So this secondary chromosome is not present in all chromosomes. Only there are certain specific chromosomes known as marker chromosomes where only these are present. Suppose you take the case of human being. In case of human being, there are such 5 chromosomes having secondary constriction, like the chromosome number. The chromosome numbers are usually the chromosome numbers you will find the 13, 14, 15, 21, 22. Only in these chromosomes you will find this secondary constriction. Now the secondary constriction, it is also known as the nuclear organizing region. This is also known as the NOR. Why NOR? That means it is a nuclear organizer region, because it is this region which is responsible for synthesis of a nucleolus. It gives rise to nucleolus. And in this region, there is the gene rDNA is there. The gene rDNA is there, which transcribes 28S, 18S and 5.8S rRNAs. RNA. That means in case of eukaryotes, the 28S, 18S and 5.8S rRNAs, they are transcribed only in the secondary constriction part where the gene rDNA is present, and is present in a repetitive manner. How many times repeated? Maybe 500 to 5,000 times it may be repeated. Or you can write simply it is repeated several times. And the part of the chromosome which is beyond the secondary construct, that means the knob-like structure. The knob-like structure is known as satellite. It is known as satellite or also trabant. And the chromosome is known as SAT chromosome. Now this chromosome is known as SAT chromosome. That means in case of human beings there are 5 SAT chromosomes: 13, 14, 15, 21, 22. Now this SAT chromosome, the full form is not satellite chromosome. The full form of SAT is sine, acid, thymonucleinico. Sine, acid, thymonucleinico. That means there is a without any thymonucleic acid. That means without DNA. Because thymonucleic acid is not present there. So this is the SAT chromosome, and this specific, and this SAT chromosomes usually known as marker chromosomes, because the chromosome in which it is present, always it will be present there. It is not changeable. So that is why it is also known as the SAT chromosomes are also known as marker chromosomes. Then the tips of the chromosomes, these are known as telomeres. And one chromatid has 2 telomeres. That means the entire chromosome has 4 telomeres. This side 2 and that side 2 telomeres. Now the telomeres are usually non-sticky. They are non-sticky. That means they do not allow attachment between different chromosomes, so that each chromosome keeps its identity. It is non-sticky. And at the tips of these chromosomes, there are some nucleotides are present more repeatedly than others. And particularly this guanine and thymine, these two are present repeatedly. Particularly in case of human being, It is TTAGGC, sorry, GGG and AATCCC. This is repeated around 500 to 5000 times only at the telomeres. That means there is a repetitive DNA, and you see this is a there is a G and T, they are present repeatedly. Now, what is the importance of this telomeres? Why these repeated sequences are present? When the DNA undergoes replication, and mostly during cell division, in each replication from the terminal end, 200 base pairs are deleted. In each replication, 200 base pairs deleted. So, if for several times it will undergo repeated as happens in case of cell division or in case of the meristematic cells where mitosis takes place, that means after several mitotic divisions, the telomeric end will be completed and the chromosome will be very short. In that case, the DNA's enzyme that will attack the main DNA. That is why how these base pairs will be each time that will be again synthesized so that the terminal end will not be decreased. It is possible because at the terminal there is one enzyme known as telomerase. Telomerase. The telomerase is a reverse transcriptase enzyme. It is a reverse transcriptase enzyme, and this enzyme is responsible for synthesis of these base pairs at the telomeric ends. As a result, the DNA enzyme cannot attack the main DNA. So, this is the structure of the DNA. There are two longitudinal consist of two chromatids. Centromere separates the smaller and bigger one. The smaller one is P and the larger one is Q. In the centromere there are kinetochores and which draw the chromatids during cell division. And in some chromosomes there is a secondary constriction where which is also known as ANOR, nucleolar organizing region, because that synthesizes nucleolus and it synthesizes all these different types of rRNAs. Then also you know what is SAT-chromosome and the telomeric ends and what is important. This is the morphological structure. Now we will discuss the chemical composition of the chromosomes. Chemically, the chromosome consists of nucleoproteins, where the nucleic acids and proteins are in the ratio of 1:1. Chemical composition, usually the nucleic acids and proteins, they are present in the ratio of 1:1. And the DNA is around 40%, RNA is 10%, that means 50%. Similarly, we will find that the histones are 40% and non-histones are 10%. That means these are 50% and these are also 50%. That means almost in the ratio of 1:1. Now, what is the importance of this basic histone proteins? Histone proteins are the low molecular basic proteins. The histones, they are low molecular weight, that is low molecular basic proteins, and they are rich in two amino acids, that is lysine and arginine. So, what is the importance of this basic protein histones? Usually, the DNA is negatively charged because of the phosphate bonds formed between nucleotides. So, the negative charge of the DNA is neutralized by histones. by histones. This is also a very important point. The negative charge of DNA is neutralized by histones. But again, another important thing. In case of prokaryotes, the histone proteins are not there. So in prokaryotes, but DNA is there. Then how the negative charge is neutralized? So in prokaryotes, it is by another protein, that are polyamines. In bacteria, the negative charge is neutralized by polyamines. And these histone proteins, are of 5 different types. There are 5 different types. 5 types. So what are the 5 types? H1, H2A, H2B, H3 and H4. These are the 4 types of histone proteins or basic proteins present in chromosomes, and the main function of basic proteins is to depress genetic activity, while acidic protein they trigger genetic activity. So these are the different chemical compositions of the chromosomes, and this is the morphological structure. The remaining part, that what is the ultrastructure of the chromosome, that we'll discuss in the next video. So thank you very much for watching the video. I hope you have a very clear understanding about the chromosome structure and several concepts relating to this chromosome structure. Wish you a very bright future. God bless you.